So, in all of these examples, we've looked at what we call simple Mendelian inheritance, where there's a clear dominant and recessive allele. That's not always the case in the real world, unfortunately. So, we're going to look at a couple examples of deviations from simple Mendelian inheritance. The stuff going on behind the scenes is still effectively the same, the way the alleles are moving, but how they interact with one another is going to be different. So, we're going to talk about incomplete dominance. This is where heterozygotes show what is called an intermediate phenotype. I'll show you an example of that. Codominance, this is where two different alleles could both be expressed in an individual. We'll see that. Multiple alleles, this is many different alleles for the same gene in a population. So we've been looking at where there's one and another allele, but what if there's like 5 or 6 different alleles in the population? How do those interact? We haven't discussed sex chromosomes. For plants, there generally are not sex chromosomes. It's very rare for plants to have sex chromosomes. So we haven't discussed X-linked or Y-linked traits. We'll look at X-linked here. A mutant allele could be located on the X chromosome of a human. So this will show sexually linked patterns of inheritance. And then we also have the possibility of certain mutant alleles in an essential gene being lethal. How does that affect the ratios that we're going to see? So let's look at these alternatives from our normal dominance and recessive. Okay, first up, incomplete dominance. All right, this is real dangerous if you don't pay attention, but there are some times where the number of dominant alleles can affect the phenotype. So sometimes heterozygotes have an intermediate phenotype, and we see this in some flower colors. Here we see a monohybrid cross, homozygous dominant red flower crossed with the homozygous recessive white flower. Unfortunately, in this case, the F1s show up intermediate. Now you're going to say, well, doesn't this prove blending? Not really, because the alleles are still discrete units. It just is how it's expressed in there, how the pigment is expressed. So there's just half as much pigment being expressed in here. So the heterozygotes will show up as pink. So our F2 ratio would be 1 red to 2 pink to 1 white. So the phenotype ratio will actually match up with the genotype ratio in an incomplete dominant system for the F2s. Okay, that's one possibility. What about this one called codominance? So codominance, there are 2 different alleles in an organism, but instead of one being dominant and one being recessive, they're both expressed at the same time. Our example for this is ABO blood type. Okay, so on your red blood cells, there can be an A antigen. There's a gene that codes for this A antigen protein. There is also a different version, the B antigen. So these are 2 different alleles. A person who is a heterozygote who has the A copy and the B copy will express both the A protein and the B protein. So you could be type AB blood because you're expressing both of them. Also, as a further kicker, there's kind of a third allele in here, a null allele basically, that makes no protein on the outside. So that's our group O. So let's look at the possible genotypes for different blood groups. So for blood group type A, you could be homozygous for A, or you could be heterozygous, A and null allele here, the little I, that makes nothing. So you just only make the A antigen. For B, BB or BI, little I, those will both only make the B antigen. If you're AB, you have an A allele and a B allele. Both of those get expressed. You make the A antigen and the B antigen, so you are type AB blood. And if you have both little i's, you make no antigen, and you are group O. Now, the weird thing about our bodies, just as a side note, is if you have group A blood, you make antibodies that will destroy group B blood. So if you have type A blood and you get a transfusion of type B blood, it will actually make you sick because your body will start fighting that blood. So if you have A, B, you're good. You can take either A, B, or O. Unfortunately, if you're type O blood, you will fight off type A blood and type B blood. You can only accept type O blood, but you can give your blood to anyone. So, pro tip, find someone or marry someone in that has type O blood, right? Then you can steal their blood from them. They will not be able to take your blood, but you'll be able to get theirs, right? You also have someone with a blood transfusion. It's a joke. It's a joke. Don't marry somebody based off their blood type. Okay. So, we have here codominance. It gets a little more complicated when we have multiple alleles. We are not going to go into the details of this example here, but I'm going to show you the broad strokes of it, and we're going to learn a couple of terms in here that are important. So when there's multiple alleles, there are many different alleles for a gene in a population. So in a diploid, there could only be 2 types, but in the population, there could be more. In this case, we have 1, 2, 3, 4 different alleles for this trait. That means that there's not just one dominant and one recessive. Their interactions could be much more complicated. This brings us to the concept of what we call the wild type. The wild type, our definition is going to be the most common form found in nature. Depending on the organism, there are different ways of depicting this. In some animals, we use the superscript plus here to show that it is the wild type allele. So in this case, this rabbit with brown fur is big C, big C. That's the wild type, the most commonly found in nature. We might see variants of this, all other phenotypes deviating from the wild type. So here we have kind of this chinchilla color, this Himalayan color where it's like white with dark embellishments, and then the full albino where there's no pigment at all. The different combinations here of the different alleles lead to these different things. This is often affected by something called gene dosage, how many copies there are of these different things, and how much expression there is of these phenotypes in different situations. I'm gonna give you a kind of crazy example. Generally, in these cases here, like this Himalayan one, where we see like the black on the nose and the paws and things like this, this is where there is expression of pigment in cooler areas of the body. So you'll see in the ears, in the nose, the extremities, there's pigment, but in the warmer parts, there isn't. That's weird, right? Like that's weird expression going on. So that's like a temperature sensitive expression there. Multiple alleles is much more complicated. If you move on to 300 level genetics, you might see some examples of this, but they are complex. So why do I bring up this wild type? Well, wild type does not necessarily mean dominant. So here I show you an example of a fruit fly. Here's a fruit fly head. If you're not familiar with it, this is Drosophila melanogaster. It's got its little mouth parts down here, and here are its little antennae, and here are its eyes. Okay, here's normal wild type antennae. Well, there is a dominant mutation that if you change one letter in one gene, the antennae will be switched. Instead of developing as antennae, they will develop as legs. This mutant is called antennapedia, and it is dominant. It is not the most common form in nature, right? Because these individuals, they don't live very well. They can't sense their environment properly. So this is really interesting, this antennapedia, right? A single change to one gene, and instead of making antennae, you make legs. That's crazy. These are called homeotic mutants. These are some of my favorite things ever, right? And indeed, several years ago when I was teaching this at another institution, one of my students who is a fantastic artist, he also thought antennapedia was cool, and his unique art style, he drew me this drawing of instead of antennae, they are making legs here. So if you like Ren and Stimpy style artwork, he does very, very cool work like that. So that's my fun little side art project that someone gave me. So let's look at another example. So that was showing that just because the wild type is there doesn't mean it's dominant. We also have multiple alleles can be involved in things like drug resistance. Plasmodium falciparum, it's a eukaryotic parasite spread by mosquitoes. The parasite, the eukaryote, causes malaria. The mosquitoes spread it from individual to individual. The malaria, the plasmodium, has a complex life cycle, but there are infectious stages for humans. Those stages are haploid. Many different drug resistance alleles have evolved and been shared between different populations. So, in the case of malaria, though, because it's haploid, it only needs one copy of those drug resistance genes for it to be resistant. So we are seeing like sulfadioxin and chloroquine resistance in malaria, which is very, very difficult. We spend a lot of money trying to solve the malaria problem. Generally, in tropical regions, we see malaria quite prevalent there. We are actually seeing also different resistance alleles in different regions around the globe. So it's taken as little as a decade for resistance to drugs to evolve in these populations, which is a very concerning development. So let's look at X-linked or sex-linked traits. In many animals, the sex of the individual is determined by sex chromosomes. In fruit flies, at least one of the systems is if you have an X chromosome and a Y chromosome, you're a male fly. If you have two X's, you are female. This isn't the only way. I believe males can also be formed if you only have one X chromosome. That's kind of weird, but anyways. So let's get some terminology out of the way. These sex chromosomes are sex chromosomes. They're specific and they have certain genes that lead to the sexes there. Autosomes are all the other chromosomes that are in there. They're the non-sex chromosomes. So you have sex chromosomes and you have autosomes, which are the rest of them. In humans, the system is two X chromosomes, that is female biologically, an X and a Y chromosome, that is male biologically. Males are what we would call hemizygous, right? They only have one copy of the X here. That is important. If you see a mutant allele on the X chromosome, for males, they only need that one mutant allele to express it because they can't be heterozygous. They only have one copy. So we see different patterns between male and female for X-linked traits. Obviously, Y-linked traits, you're only going to see those in males because if you have the Y chromosome, you develop male. So in Drosophila, white eye color is recessive, but it's X-linked recessive. So a female that has white eyes is XW for white eyes. These are little w's, by the way. Here we have a male that has red eyes, X big W. So when we do the cross, we have to look at this. Here we make some male offspring, but they get Y's from the male and X's with little w's from the female. So both the males are going to be white-eyed here, while both of the females are going to get this dominant allele from the male parent, and they will both be red-eyed. So instead of seeing a 3-to-1 ratio like we would expect, we're seeing a 1-to-1 ratio here. So what are some human X-linked traits? You may have heard of some of these. We have things like color blindness, generally red-green color blindness, hemophilia, muscular dystrophy. These are all X-linked. So hemophilia is where you lack a clotting factor, and if you get a cut, you bleed and don't clot very well. So because sons can't be heterozygous for X chromosomes, they're going to show the phenotype much more often. So an X-linked recessive mother, if they're affected, all of their sons will have this, regardless of the father's condition, right? Because the mother gives the X to male offspring. So here we have a heterozygous or carrier mother. She has one recessive allele and one dominant allele on the X. Here's the father. Let's look at the male offspring. Well, for male offspring, the father's giving the Y. So here we have the two Ys. And in this case, the mother could give that chromosome, unaffected son. But if she gives this one, she would have an affected son. But let's look at the female offspring. Well, here the father is giving an X and the mother is giving the affected X. But because they're heterozygous, because they have two copies of the X chromosome, the daughter is unaffected here. So basically, any trait where you see sons being more often affected, that could be human X linked. So let's look at a pedigree here for an X-linked recessive trait. So we have males are squares, females are circles. Okay, filled in is affected. Look at this cross here. We see unaffected females, affected males. And then this here, affected male, affected male, affected male. Here we do have an affected female, but to get that, we had to have two affected parents there. So let's work out what the possible genotypes could be. Well, this affected female is going to be little x, right? Both recessive alleles. This father will have unaffected X, so big X, and a Y chromosome. For the males, the Y gets passed on and the affected X gets passed on. This mother's male offspring will always be affected by this condition. The females are unaffected because the male is passing on the unaffected X chromosome. Here, all of their children will be affected because there are only affected X chromosomes in there. And this one, we have a carrier female here. She's passing on that affected X chromosome to one of her male offspring. So 50% chance for the male offspring. All right, so our last example of deviating from simple Mendelian. Lethal alleles. These are mutant forms of an essential gene that basically, without the proper copy, the organism won't live. If this is recessive, it can persist in the population, right? Homozygous dominant will be fine and heterozygotes will be fine. Only the homozygous recessive individuals will die from this. Generally, we would then see a genotype ratio of 2 heterozygous for every 1 homozygous dominant, since the homozygous recessive don't survive. There is the possibility of dominant lethal alleles as well. This can happen if and persist in the population if the lethality happens after reproductive age. Huntington's disease is an example of this. Huntington's disease is an example of basically a disease that leads to nervous system decay. This does not become lethal until the generally late 20s, early to mid 30s for most individuals. So people could have reproduced before then. So here's a pedigree. We have an added little bit where we have a slash through deceased individuals. And we can see their age here where they started to show the onset of the disease. Here, this individual died at age 34. So they obviously reproduced. They had 1, 2, 3, 4, 5, 6 offspring before they died from this disease. One of their offspring died at age 33. This one made it to 38 and had 3 children of their own. One of them died at 21. So you can see that this lethal allele is persisting in the population. And this individual may not have even known that they had it until we see the offspring. So this brings up interesting and difficult ethical questions about what do you do here, because you're going to pass on this dominant allele potentially to your offspring. That's a tough question to ask people. So there's the whole concept of genetic counseling to give people the probability of having affected offspring. All right, so to finish up, Mendel used probability rules to explain all of this. Probabilities are mathematical measurements of likelihood. How likely is something to happen? And we have empirical probability, what we observe, versus theoretical probability, what we expect. So we could observe something a number of times over the total number of opportunities. versus times expected over total opportunities. That would be the theoretical side. So probability is a range from 1 to 0. 1 is when it always happens. So if you had 10 opportunities and you observed it 10 times, that's a probability of 1, right? 10 over 10. If you had it 0 times and you had 10 opportunities, that's 0 over 10. That's 0. It never happened. So probability can be used to explain these different things. Let's look at how we utilize it here. And it's really critical because the probability rules have two rules. We have the product rule. The product rule can be applied to the independent transmission of characteristics. So let's give a generic example here. We're looking at rolling a die and flipping a penny at the same time. The product rule is the probability of two independent events occurring together and can be calculated by multiplying individual probabilities together. What's that mean? Well, let's give an example. What's the probability of rolling a 6 and flipping heads? You'll notice occurring together, I've underlined and because we want a 6 and heads. Okay, what's the probability of that occurring? Well, on a 6-sided die, you have a 1 in 6 chance of rolling a 6. On a 2-sided penny, you have a 1 in 2 chance of flipping heads. So to get a 6 and heads, you take 1/6 probability times 1/2, you get 1 in 12 chances of that occurring together. So this could be like, what's the probability of getting a green seed color and a wrinkled seed shape together? We might want to know that, and we would use the product rule of those two things. The sum rule, on the other hand, is the probability of the occurrence of one event or the other event. These are two mutually exclusive events. It's the sum of their probabilities. So rolling a die and flipping a penny at the same time, what is the probability of rolling a 6 or flipping tails? Again, we have a 1 in 6 chance of rolling a 6, but now we add it to 1 over 2 chance of flipping heads. So it's this or that. Here, we can simplify this as 4/6 because of the way the math works. So that's 2 in 3 chances of getting a 6 or flipping tails. We have a 2/3 probability of that happening. How could we use that? Well, what is the probability of having at least one dominant trait show up in the F2 of a dihybrid cross, right? What's the probability of having a purple flower or a yellow seed in a plant? We could use the sum rule for that. All right, review question. If you found a new genetic disease that affected males much more often than females, you might assume the allele controlling this disease is, pause the video, think about this. This would be X-linked most likely when you see it showing up in males much more frequently. It's generally X-linked because males only have one copy, so they only need one affected copy to show the disease. All right, if it was autosomal dominant, we would see it at a 50-50 rate between males and females. Same things with autosomal recessive. There would be no leaning towards male or female in any way. All right, that was a doozy. So we've talked about traits. They're controlled by genes on chromosomes. Different versions of these genes are called alleles. Phenotype is the expression of a trait that we can observe, while genotype is the underlying alleles behind the scenes controlling this. In diploids, there are 2 alleles. You can be homozygous, 2 copies of the same allele. So you could be homozygous dominant or homozygous recessive. Heterozygotes have 2 different alleles in there. We talked about the test cross, where we're taking a dominant phenotype individual and we want to figure out their genotype. So we cross them with a homozygous recessive individual. We know what their genotype has to be. And then we can look at that ratio to figure out what's the genotype of the dominant phenotype individual. For organisms you can't easily cross, you have to rely on a family history pedigree. Causes some problems sometimes. We looked at some alternatives to simple dominant and recessive, incomplete dominance, codominance, multiple alleles, X-linked, and lethal alleles. Then finally, we finished up with the 2 probability rules, the product rule, finding the probability that one thing and another thing will occur together. The sum rule, on the other hand, finding the probability that one thing or another thing will occur. All right, that's it for 12.2.